Hi, everyone, and thanks for joining us. I think we will go ahead and get started as the last few people roll in. Thank you again for joining Prairie Rivers Network for our community briefing on pesticide drift in public spaces. I'm Devin Walker, and I'll be helping out with the presentation today and helping out with the Q&A at the end. Just a note that this webinar is being recorded, and if you have any questions along the way, please feel free to put them in the Q&A down below, and we'll have some time at the end to go ahead and address those questions. Also, we will be sharing some important links throughout the presentation, and then again in the follow-up email that you will receive in the next day or two. I will go ahead and hand it over to you, Kim.
Thanks, Devin. Thanks for being here, everyone. Today, I'm going to talk about our addendum to Hidden in Plain Sight, pesticide drift in public spaces. But before I do that, for those of you that aren't very familiar with Prairie Rivers Network, let me give you a little bit of background about our organization. Our mission is to protect water, heal land, and inspire change. We use the power of sound science, the law, and community action to create that change. And we've been around we're about to celebrate our 60th birthday, and we've have had a wide range of program areas throughout the history of our organization, but a constant has been the work of water that has flown through the decades of our work here in Illinois. Today, we focus on renewable energy, climate change, water quality, water quantity, pesticides, the protection of biodiversity, river health, and environmental policy. So as the Director of Ecological Health, I lead our team that works on the protection of biodiversity in the state of Illinois. And so some of our team works on river health and resiliency in a changing climate. Some of them work on pesticides along with myself, and others work on wildlife habitat and focusing specifically on pollinator habitat. So let me give you a little history of the issue of drift before we start going into the report. Back in about 2016, 2017, Prairie Rivers Network started getting calls, emails, and letters from concerned citizens that were worried about the appearance of curled and cupped and injured leaves on their trees and plants. And we knew that these were symptomatic of off-target herbicide injuries. So together with some of our dedicated volunteers and members, we started asking questions. Are there any agencies across the state that are looking at the prevalence and the severity and the distribution of herbicide injuries across the state? And what we found was there were not, nor was there any agency that was monitoring pesticides that are drifting through the air. So with this in mind, many of us came together and we developed the Tree and Plant Health Monitoring Program, which really took off officially in 2018. And it was at that time very much on a shoestring budget, very heavy on volunteer and member assistance and getting this off the ground with minimal funding. And over the years, we've been able to grow it. People are deeply concerned about this issue, and we continue to monitor this because we care deeply about this issue. So we use pesticides for many reasons, and they are fantastic tools. And when I use the term pesticides, I mean fungicides that target fungi, herbicides that target plants, and insecticides, which can target invertebrates. There's rodenticides, nematicides, many different types of pesticides. For the purposes of this talk, we are largely focusing on herbicides and fungicides. But we use these chemicals in the control of broadleaf weeds and turf management, whether it's a golf course or a backyard, to manage unwanted woody vegetation or plant growth in utility rights of ways. We use them to grow commodity crops and food crops, and we use them to manage the places that we care deeply about to help eradicate invasive species in our prized natural areas. But one of the leading uses of pesticides in the state of Illinois and throughout much of the Midwest in the country is agricultural production. And in Illinois, that is because 75% of our land, or approximately 27 million acres, is dedicated to agricultural production. And that can be row crops, that could be specialty crops like peaches, apples, pumpkins, grapes. That could also be fruits and vegetables, which is another type of specialty crop, or pasture lands and grazing lands. But the majority of that 75% or 27 million acres is planted in row crop agriculture. And the majority of that is planted with herbicide tolerant soybeans and corn and other crops. And when I mean herbicide tolerant, that means they are genetically modified to be resistant to herbicide spray so that growers can plant these crops and spray herbicides over the top of growing plants, killing the weeds, and not harming the plant itself. And so this has really changed, this innovation has really changed how we grow crops in our country, particularly these commodity crops. And where historically we would use herbicides early in the season for weed control prior to planting, and then use light tillage or cultivation or other methods of weed control throughout the growing season. Now we can use herbicides for weed control throughout the growing season over the top of growing plants. And for some species, it's to a specific life stage that you can apply over the top. For example, in soybeans, there are cutoffs for that. Additionally, there are fewer fence rows in our landscape. The equipment that we're farming with is bigger, our fields are bigger. And all of these things have kind of helped perpetuate this problem of overuse of herbicides and over-reliance on herbicides. And we also have an agricultural system that has much less crop diversity than historically so. So we might have my grandparents per se, or your grandparents may have been farmers, or even your parents, where they had multiple crops that they rotated on their farmland. And this may have been, vegetables or pumpkins or corn, soybeans, wheat, alfalfa, other types of crops. And what that diversity does is it interrupts pest cycles, whether it's a disease, whether it's an insect, or whether it's a weed. And when you don't have that interruption and you're using the same types of weed control over and over, problems arise, such as resistant weeds. So let's talk about drift. Drift, pesticides move through the environment in many ways, but when we're talking about drift in this talk, we're talking about drift through the atmosphere, through the air. And here we have a crop duster that is applying a pesticide over the top of corn. And I'm using this picture because it's a really great example of the particles that are in the air at the time of application. And you can see those particles and you can see how they are moving in the air column. Now that is an example of particle drift when pesticide particles are moving at the time of application. They can be carried by the wind. If it's a really gusty day, they can be carried long distances. Now, let's think about that plane has landed. This crop has been sprayed. The next day, 3 days later, 5 days later, we get some favorable weather. There's another type of drift that can occur, and that's called volatilization or secondary drift. And that's when those pesticides volatilize or essentially evaporate off of the plant and soil surfaces, go into the atmosphere, and they can move in the air column off target and land, causing injury to non-target plants. And when we think about the issue of volatilization, I think it's important to realize that we are applying many of these pesticides to thousands and thousands of acres, and oftentimes in a similar time frame. And weather patterns, we may get a regional area in the state where we have warm weather for a week, and that can be perfect temperatures and conditions for volatility. And that's where we might have landscape level injury occurring across the state and throughout the Midwest. And we have seen that before with the use of dicamba, particularly back in 2018, 19, and 2020. So what do these symptoms of drift look like? Well, the easiest type of pesticide to observe symptoms of drift in are herbicides. Herbicides leave a physical marker on the plant. And when we're looking at them in our monitoring program, we're looking at cupped and curled leaves. Leaves may be disoriented, sideways, like twisted at that petiole, upside down, or even drooped. They may also have irregular margins where the edges of the leaf may be scalloped, wavy, or pointed in an exaggerated manner. They may also just be deformed generally and stunted where they haven't reached their full size. And then there's a common symptom called chlorosis or yellowing of the leaf. And that can be the entire leaf, or it can be yellowing along the main veins of the leaf. And this photograph here is a photograph of leaves from our state champion post oak a few years ago. And it pretty much has all the symptoms that I just listed. And these symptoms aren't something that we've discovered. They've been a diagnostic tool for looking at herbicide injury for a very long time and are in many publications in Illinois and across the United States that are widely used. So in our monitoring, our tree and plant health monitoring program, when we go to a site, the first thing we're doing is we're ruling out any direct application exposure to the plants that we're seeing. And that means, what are we near a right-of-way? Was the landowner or manager using herbicides for cosmetic weed control or for turf applications? Are there broadleaf weeds in the grass? If we see clover and chickweed and other broadleaf weeds, then that's a good sign that they are not using turf management or chemical turf management. Additionally, are there symptoms present across multiple species and families? And this is really important. When we're looking at a site and we're going to look at redbuds, dogwoods, maples, box elder, poison ivy, vining grape, ground plants such as rosin weed or cultivated plants in people's yards and gardens. And we're looking to see if there are symptoms across these multiple species and families, because if there are, then that's a very strong indication that what we're looking at is drift. Fungal infections, diseases, and pests are not going to hit or impact every species and every plant type at a location. That's a good way to rule those things out. This is just a brief overview of our monitoring form. We collect information on where we were, who's doing the observations, the date, a little bit about the weather. Then we also record information about the species that we're monitoring, the number of those species that we've monitored at a site, as well as the symptoms that they're demonstrating, whether that's the cupped, curled, irregular margins, et cetera. And then we rate those symptoms. 0 is there's no injury. 10 is whole plant death. At the bottom is where we have some site ecology here, and this is on our first page. We typically monitor anywhere from 9 to 15 species at a site. We're walking through the entire site, or as much of it as we can, and we're observing plants in the overstory, understory, and at the ground layer. So in 2024, we released a summary of our monitoring efforts from 2018 to 2023. It was hidden in plain sight. And in that report, we have a page for by the numbers. And I've updated some of those numbers with the help of Martin Kemper, my colleague in this work. And from 2018 to 2025, we've performed a total of 1,028 monitoring visits to 304 monitoring locations or sites, and 64 of those sites we have visited for 4 or more years. We've also documented symptoms in 315 species, 256 of which are native, and 71 plant families. And you can see here, this is a photo of our white oak estate tree, our state tree, with herbicide injury. Of those species, 109 of them are trees, 18 of which are our native oaks. And that's really important because oaks are keystone species in our oak hickory forest, and 56 of those species are shrub and woody vines like grape and buttonbush, etc. And then we've documented symptoms in 149 herbaceous plant species, whether those are cultivars or prairie plants or other woodland ground plants. Since 2019, we have not monitored a single site that does not have symptoms throughout. So what are the consequences of chronic long-term injury? Well, we're seeing a lot of thinning tree canopies. We're also seeing epicormic branching, where trees are pushing out leaves along their main branches and trunk. That's sort of their last-ditch effort to produce leaves to make food for themselves. We're also seeing a lot of branch dieback, as you see here in this white oak. And unfortunately, we are documenting tree death in many of the areas that we've been monitoring for many years. So what do we know about the ecological impacts of herbicide drift? Well, the literature tells us a few things. First of all, exposed plants produce fewer flowers. Those flowers are visited less frequently by pollinators. And plant tissue that has been exposed to herbicides may have lower nutritional content. And there have been studies, even one that's been performed by the Illinois Natural History Survey, that show that caterpillars that eat vegetation that has been contaminated have lower biomass and greater mortality. All of these things can add stress and more work for insect feeders, like our migratory birds. And they can lead to potential disruptions in our plant and pollinator networks, which is deeply concerning. Additionally, we've seen at many of our sites that exposure can result in reduced fruit and nut production, providing fewer resources for wildlife and increasing the stress they have to encounter for food production or for gathering food. And so this isn't just for ecosystems. When in 2018 and 2019, when dicamba injury was widespread across Illinois and much of the Midwest and South, many of the orchards, including ones down in Southern Illinois by me, had smaller fruit. Fruit would drop early. They lost a lot of their crops because of herbicide injury. So it's not just impacting fruit and nut production and wild plants, but also our cultivated plants. And it's important for us to remember that these drift injuries are additive to our current stressors, such as habitat loss, invasive species, whether they're plants or animals, diseases and pathogens, fire suppression, and extreme weather, drought and floods. So oftentimes when we're monitoring and we're looking at drift across multiple species and plant types, we're also seeing pressures from insects, pathogens, weather, and it is concerning because we have a poor understanding of what these additive impacts are. And what do they mean for our communities? So, we're summarizing this, and we have all of these stressors to tree health: pesticide drift, invasive pests, disease, etc., which lead to declining tree conditions, that canopy dieback, reduced growth and mortality, and this loss of ecosystem services, less shade and cooling, changes in understory plant communities, reduced water and air clarification, and losses in wildlife habitat. There's also environmental changes. We have higher temperatures in our community. There's a lot of research that shows that when communities have dense tree canopies, they are cooler than without. They also reduce flooding and protect us from soil erosion and poor water quality. So there are real community and economic impacts associated with the impacts of pesticide drift, and they can impact our health, our wallet. Taking down a tree is costly, whether you're a park district, a state park, or a private landowner. And there's a reduction in quality of life. Not only do we witness the decline of the health of the trees in our communities, but many of the people that we worked with over the years are deeply concerned about the impacts to the places that they love and hold dear, whether it's a place that they visit or a place that they manage. And that impacts their own quality of life because they have to see it year after year. So one of the things that we have done in our monitoring to help us understand, we're seeing these symptoms, some of these places that we're monitoring, like the Kaskaskia macrosite forest, is one of the largest contiguous bottomland forests in the state of Illinois. And, we're out there in the middle and we're seeing injury from drifting pesticides, and we're taking a leaf sample. And we wanted to have a better understanding of what's the closest potential source that could be causing the symptoms that we're documenting in our monitoring. And so we started ground truthing it, and we're using, visual sight by knowing the area and talking to the landowners, but also using satellite imagery such as Google Maps to measure the distance of the furthest point into that property where we monitored and documented symptoms to the closest potential source, be that a golf course, an agricultural field, or whatever other source of pesticide use could be nearby. And that is not necessarily and most likely not the actual source of drift. It may be another source or multiple other sources, but that helps us give an idea of where's the closest point this could come from. And this graph illustrates that. And if you look at the top line, several of these monitoring visits hit that 3,500 feet to the closest potential source. That's a long distance for some of these pesticides to travel. So we have the monitoring, but we also have a tissue sampling component of our program. And we have developed and follow a very strict sampling protocol. And we have our samples analyzed at certified labs. We've used South Dakota Agricultural Laboratories for many years, really throughout the majority of this project, with the exception of the first year and some of the samples. And we really trust their methodology, and they're very easy to work with in a variety of ways. But having tissue samples analyzed helps us have a better understanding of what types of pesticides are moving. We're not definitively able to say that this pesticide is the one that caused these injuries because we are out there collecting these samples at a site without any knowledge of when applications occurred. We're out there, we're documenting, we collect a sample. So we don't know exactly when that drift occurred or what that applicator was using, but this will hopefully give us an idea based on the analysis that the lab runs of what pesticides are going through the air. And so here's some of the results from our tissue analysis. 2,4-D is a pesticide that is frequently detected. It's a growth regulator herbicide. It's widely used in turf management as well as agricultural production. And we've gotten detections of 2,4-D at 3,000 feet from the closest potential source. Dicamba, I mentioned that earlier. It's known to be very volatile. It can move long distances. It's widely used in agriculture. It's also used in turf management. And we've detected it also at 3,000 feet from the closest potential source. And I want to note here that we have 85 detections of dicamba. However, dicamba is one of those pesticides that can be tricky to detect because once you're seeing symptoms, that chemical has most likely already biodegraded. So the fact that we have grabbed leaf samples with detectable levels of dicamba in that is showing us that either that herbicide was causing those injuries or other herbicides were with it causing those injuries, and we just happened to catch it. Atrazine, widely used in corn, again, 3,000 feet from the closest potential source. Glufosinate, a widely used herbicide these days, and it is one that had us scratching our heads for a while. It's not known to be volatile and move long distances, but we were getting these detections, 3,000 feet away from the closest potential source. And our only, explanation for this or assumption is that someone was applying these pesticides or glufosinate in combination probably with others in a tank on a windy day, and it drifted long distances during that event. This majestic creature is our state champion post oak. It's located in Washington County. This tree we've been visiting and monitoring since 2017 and taking tissue samples on it since 2018. And the people that you see below in this photograph are some of the key volunteers and dedicated people that have been working on this issue with us at PRN for the past 8 years. And we wouldn't have this monitoring program or any of this data without their help. So when we visit this tree, this is one of the few sites that we visit multiple times a year. These tissue samples are extremely expensive. And we have our time and the travel to visit these sites and do the monitoring. So there's only a few sites that over the years we have visited multiple times throughout the growing season, and this tree is one of them. This whole field is one of them. And we've taken roughly 3 samples a year from this tree and been able to document that it experiences multiple drift events throughout the growing season, year after year. So it's under chronic stress. And, this is a photo of the canopy from a couple years ago. Not every year is the same. Some years are worse, some years are better, but this tree is under stress. It is experiencing branch dieback. Its canopy is thinner. Its leaves are cupped and curled and deformed every single year. And it's our state champion. So I'm about to take you on a tour of the research that we did last summer, but first I want to cover a couple of things to explain. When we were evaluating our next steps after Hidden in Plain Sight, we had questions that we wanted to try to find answers to. And some of those questions are, how frequently are drift events occurring in multiple places across the state? What pesticides are moving during those times? And how can we best capture it? And so we designed a study with the help of some advisors on how to answer some of those questions. And we chose we went through a couple scenarios of ways we could get more data and more information about what pesticides were traveling. We thought about air samplers. They're too expensive, they're obvious, they're easily vandalized or removed, and it's not something that we can replicate at each site. We thought about testing surfaces, but that's really labor intensive, can be complicated to analyze, and there's just a lot of unknowns associated with that. And then we thought about monitoring bands that have typically been used for personal chemical exposure analysis. And those are typically worn by an individual for about a week and then sent to a lab to be analyzed. The use of having them out in the environment is fairly novel, but it's an inexpensive experiment to run. And that's what we decided to go with. We ordered monitoring bands that were prepared to be deployed out in the environment from a company called My Exposome. They are actually the ones that developed this methodology of the personal monitoring devices for chemical exposure. Dozens and dozens of papers have been published using these bands for chemical exposure detections. So we wanted to pair those bands out in the environment with our leaf monitoring and the analysis of leaf tissues to see if we could gain a better understanding of what was moving through the air. We could place multiple bands at locations without risk of them being vandalized or stolen if we lost one or two. We weren't too worried about that as opposed to a very expensive air sampler. So here we have a photo of our bands in a tree. They were placed in the same trees that we collected leaves from, and we collected leaves and put bands in 2 trees at each site, but we only analyzed the bands and leaves from 1 tree every month at every site. What we did is we chose 10 parks and schoolyards around the state. We tried to spread them out geographically, 2 in the northern, 4 across the center of the state, and 4 down in the southern portion of the state. We did full PRN monitoring in the months of May, again in July, and in September. And we did that because leaves tend to hold on to their symptoms. Once they have injury, they may get worse through consecutive or multiple exposures, but they're going to hold on to those injuries. Herbaceous plants, they might get hit, but then they're going to continue to grow through the growing season. So the severity of those injuries, if they don't get hit again, are going to lessen. So for the sake of our time and for just, data collection, we chose to only monitor 3 times in the season. However, we deployed bands in May, June, July, and August, as well as collected tissue samples. In the beginning of May, we put out our first band and grabbed that first tissue sample. When we went back the next month, approximately 30 days later, give or take, we collected that band, put a new band in on the same tree, and collected another sample. We chose parks and schoolyards that were varying distances from potential sources. Some, of these community parks are across the street from an agricultural field or next to a golf course. We also chose them again so they were dispersed across the state. We wanted to know, is it happening more in the north? Is it happening more in the south? So we spread them out. And again, we tried to space our time of collecting and swapping out bands, collecting samples and swapping out bands to around 30 days. So we're going to start on our tour, and I'm going to talk to you a little bit about these sites. The first site that we're going to visit, I'll explain the graphic that you're going to be looking at. This is in Winnebago County, and I'm going to talk about 3 things. This graphic is one of a series of graphics that we will be using for an interactive tool that people can utilize to learn about what we did and what we found. And that should be released in a few weeks to a couple months. But this graphic is the template for some of that information. And I'm going to be focusing on the distance to the closest potential source. So this park was 3,175 feet from the closest potential source. We monitored a pin oak. Oops. And out of the total number of 26 pesticide detections, we were analyzing for herbicides and fungicides, we determined that there were 18 independent exposure events based on the analysis that we performed. And let me explain that number. So in order for there to be an independent exposure event, we had to see a new pesticide pop up in that window of time, that sampling window of time, that 30 days. And it would either be in a band or a leaf. And if that was a new detection, we counted that as an independent exposure event. If we had the same pesticide in a band and a leaf, we counted that as one exposure because they were there during the same time frame. If the level, the parts per billion of a pesticide dramatically increased from one sampling visit to the next, we counted that as another exposure. The only exception to this rule here is dicamba. If we detected dicamba in May, and then we went back and it was there in June, but it didn't increase in parts per billion, we counted that as an independent exposure because we knew the half-life of dicamba and the length of time it would reside on plant tissue was very, very short. So that had to be a new exposure. And then I'm going to highlight the pesticides with the most detections. And in this case, at this location, it was 2,4-D. You'll see over on the lower right, there's a list of all the pesticides that were detected during that summer. Now we're going to go to Kane County. And this park is 1,525 feet from the nearest potential source. It is a public open space with exercise tracks, and it is directly adjacent to an elementary school, and across the street is a middle school. There were 16 independent exposures at this location, and the most frequently detected pesticide was 2,4-D. Now we're going to go over here to Adams County. This park is about the tree that we tested was about 470 feet from the closest potential source. And there were 24 independent exposure events. Most frequently detected 2,4-D and atrazine in those samples of the swamp white oak and the band. Now we're going to head over to McDonough County. The tree in this park was approximately 110 feet from the closest potential source. There were 22 independent exposure events documented at this park, with, again, 2,4-D being the most detected pesticide. Now we're in Sangamon County. This is a park with ball fields, a walking path, a lake, and the tree we sampled is between the walking path and the lake, and it was about 110 feet from the closest potential source, and 20 independent exposure events, again 2,4-D and atrazine. Now we're in Champaign County here, and this park is located 3,700 feet from the closest potential source. We determined there were approximately 22 independent exposure events, and the most frequently detected pesticide was atrazine. Now we're heading down to the southern portion of the state. We're looking at a park that's 320 feet from the closest potential source, 22 independent exposure events, and we frequently detected 2,4-D, atrazine, and metolachlor in these samples of this bur oak. Heading over to Randolph County, another community park, 1,300 feet from the closest potential source, 21 independent exposure events. 2,4-D, atrazine, and metolachlor were frequently detected on this post oak. Heading over to Saline County, this is another community park surrounded by agricultural fields and big, beautiful oak trees and hickory. 210 feet from the closest potential source was the tree that we sampled. 21 independent exposure events, frequently detecting atrazine in those samples. And finally, down in Union County, in the southernmost part of the state, we have a park that is also part of an elementary school and a high school. It's on their grounds. And it's 630 feet from the closest potential source, a total of 14 independent exposure events. And we most frequently detected 2,4-D at this location. So when we combine this data together and we look at the number of pesticide detections per month, we see some trends here. And this graph, along the bottom are the pesticides, the herbicides and fungicides that were detected. And then the months are color coded. And what stands out immediately is 2,4-D. It's really detected at all of our sites very frequently. Also, commonly detected the herbicides acetochlor, atrazine, dicamba, glufosinate, and metolachlor. And then if you look at the fungicides, we're starting to see more of those fungicides later in the growing season, so in August and September. When we look at the sites with independent exposures, we also see some trends going on. Most of the exposures that we were documenting in our sampling were occurring in June, July, and August, trailing off again at the end of the growing season, which we would expect, and a little bit lighter, but still, quite a few detections early in that month of May. So in conclusion, symptoms of drift were evident throughout each location every month that we monitored and collected samples. There was an average of 20 with a range of 14 to 24 separate exposure events at every location. Pesticides, including those herbicides and fungicides, are moving through the environment more frequently than is widely recognized. And unfortunately, we do not have enough ecological monitoring or air quality monitoring for chemicals like pesticides. In fact, there's no air quality monitoring for pesticides that trespass onto public and private lands in the state of Illinois. And finally, there are very real, and I encourage you to talk to some of the people that have been impacted by drift at some of the sites that we monitor. They're very real and often difficult economic, social, and emotional costs to this chemical trespass. So over the years, PRN has kind of evolved in what we've done to work on this, but there's some constants, and one of them is tours. Every year we try to do tours of impacted sites to bring agency staff out, local park districts, municipal public work managers, commissioners from the Illinois Nature Preserves Commission, impacted landowners together to talk about what we're seeing, what it means, and try to find solutions. We continue to monitor and train private landowners, land managers, and agency personnel like conservationists that are working for agencies on how to monitor and document symptoms on the properties that they manage. We continue to give talks like this every year. I give them, Marty gives them, and it's our way of educating the public about this issue. We've published numerous reports starting back in 2020 with Drifting Toward Disaster, which we co-authored with the Xerces Society for Invertebrate Conservation and the National Wildlife Federation. Since then, we've also worked on our monitoring summaries in 2018 and 2019. We did a report for the Nature Preserves Commission, Herbicide Drift and Chemical Trespass on Natural Landscapes, and we published our Hidden in Plain Sight in our addendum of this most recent study. We've also been working and will continue to work at a state and a federal level on policy. We comment on pesticide registrations and management solutions that are presented by US EPA. We write joint comment letters, and we share information with partners in our other states that are impacted by this, such as Nebraska, Iowa, the Dakotas, Missouri. They're all concerned about this issue. And we work at a state level with many of our partners, the Friends of the Illinois Nature Preserves, Illinois Environmental Council, farmers, and other interested conservation groups to work on this issue. And with that, I would like to thank the many people at PRN that help make this work happen. and help us get it out there, our board, our staff, our executive director, Maggie. Martin Kemper, my colleague in this work, has just been a driving force. He's relentless, he's dedicated, he's extremely knowledgeable, and our field assistants that help us, that have been with us walking through fields and woods for years in the heat to document these symptoms, and all of our technical reviewers for the study.